So our next speaker is Dr. Marcelo Coba, who is an associate professor of psychiatry and behavioral sciences at the University of Southern California. And he studies how genetic variants affect synapse function, but uses a systems biology approach. So not just looking at individual genetic variants, but thinking about signaling networks. And so he's going to tell us about his work today. There we go, you can share. Great. And just make sure you unmute.
Yeah.
We can hear you, but we lost your slides.
Yeah. Can you see the slides? No.
Yes.
Yeah. Yes. Okay.
Yep, we see your PowerPoint. I think if you start the slideshow, we'll be able to see it. Perfect.
Yeah. Okay. Thank you. Thank you for the invitation. And I would like to. I'm going to discuss what are we doing in terms of using human models for Syngap disease. Why the reason that we're using what is good about them and what are the problems about these things. As with many on this panel, we were used to work with Syngap and other synaptic proteins within the context of mature rodent synapses. But so we're going to discuss this a bit more of what is this molecular context of Syngap within iPSC derived neurons, what is similar and different from rodent synapses, and show some what do we got with different mutations, from different patients and Syngap, and what common things what we found, what are different. But the one thing is that we need to do is that we'll try to give this molecular context and why we need to describe a bit on what do we really know in the synapses of these human mouse models. So the reason on this is that because it was some years ago, we moved forward with the lab and trying to get more translational approaches for this, not just for SYNGAP1, but for mutations in different components of the synapse. And we reasoned to do it with two main ways. One is the classical pharmacology approach, and with improving methods of stem cell and gene editing, trying to recover and improve SYNGAP1 protein levels. And Gavin gave great examples of how these things can be achieved, but I will going to mostly focus within the context of human neurons. So for any of these approaches, you are going to have at least 4 difficult things to do. It's just you will need to get a target. In this case, the target obviously is Syngap, but as Gavin showed, it can be any other component. We need a compound or editing approach, the delivery system, and a model. So what we're going to describe today, give you this. It can be a talk for any of these problems, but it's just what are we going to do with the model? What do we need to know with the model? So when we're taking this into account, our view was, okay, what do we need to know about, what do we need to understand for these human models? And one of the good things about the human models is that, well, we would like to work with a human gene, with all the regulatory elements, and with the genetic background of these particular patients. I'm not going to describe anything on genetic backgrounds, but we are heavily interested in the genetic backgrounds from different patients and the role that plays in different phenotypes. The other good thing with human models is that we can work still with single cell types. We can work with glutamatergic, GABAergic neurons, serotoninergic neurons. We are doing the three of them. Also, heterogeneous models of brain organoids, what we are doing. But I'm not going to show much data on brain organoids. We're going to describe only what we are getting with the glutamatergic type of neurons. A big problem with these models is that everybody knows these models are immature neurons. They are fetal-like neurons, and they are polycharacterized. So a main question for us was, okay, everything that we know for SYNGAP, all this molecular context that we know, that what is it doing in a rodent mature synapse, how many of these things can be reproduced, or what is similar or not similar in these type of neurons. So the idea was not to make catalogs of what is in there, but it's just a problem for us. Is I really wanted to understand the model system that we are going to work with. So unfortunately, well, I will need to give you a general background and maybe give a really great talk on some of the molecules that I'm going to describe, but I need to show you what is mostly what we know in terms of a systems approach from these rodent synapses and what do we have in these human models. So, you know, that this is a postsynaptic site, presynaptic, postsynaptic density. Many of you already know this. You have this electrodense region here that is a postsynaptic density. And in this region here, if you can isolate it and analyze the mass spectrometry, what proteins do you have there? You have more than 2,000 proteins. There's which we define a core component, core more abundant molecules, about 200. And you can see there, it's a dense core of protein-protein interactions, what we call protein interaction networks. When you try to analyze which protein is interacting with which other, you define it as a protein interaction network if you have enough data to describe which molecule is associated to each other. So you have different, what we describe, this is a cartoonish kind of thing that is different, different layers of scaffold molecules. This is the first layer of MAGOX molecules that you have DLG 1 to 4, DIRGAPs that binds to the MAGOX, DIRGAPs 1 to 4 as well, and the shanks at the bottom layer with shank 1 to 3, and each one of the others are MAGOX interacts with DIRGAPs, DIRGAPs with MAGOX and shanks, and all of them gets a dense core of protein-protein interactions. There, and SYNGAP is one of these molecules. This is an error here. SYNGAP is one of these molecules that will have interaction with different components of this protein interaction network. And I probably don't need to show this, as you already know, but you have different modules that describe functions like a RAS gap domain, different modules that is involving protein interaction, that pH domain, PDZ ligand binding. But what we describe this, when we're going to describe in terms of protein domains, is that these domains are or protein modules, it can tell you that what is the function of the protein. And we usually describe the function of the proteins by the protein domain composition. Mainly because this protein doesn't work alone, and these functions are within the context of the surrounding molecules. That's what we are describing, the molecular composition, and I'm going to show what is in these rodent synapses and in the human synapses. And more importantly, the protein composition change in a developmental manner, obviously in a cell type manner. So if we are describing that these synapses from the human mouse models are not mature, we can think about that the protein composition is not the same. If the protein composition is not the same, SYNGAP functions might not be identical. So what we need we wanted to have a pretty good idea what sort of molecules are there, what sort of molecules SYNGAP can bind to in those other synapses. So this is a more detailed view of the kind of protein domains that you have. It doesn't matter much which one is each one, but you can see all these shapes and colors are showing different domains that can bind different proteins. And SYNGAP has a collection of some of these protein domains as well. Mostly what we study SYNGAP is by only three of these protein domains. One is the RAS gap domain, the PDZ ligand, which binds to PDZ domains, and setting threonine kinase domains that are not properly from SYNGAP, but we have a abundance of data, or at least some information on what kind of kinases also relate this SYNGAP and have an effect of SYNGAP function. So I'm going to describe mostly some of the molecular composition on these two domains, RAS-GAP and serine threonine kinase within the context of the PSD. So SYNGAP is not the only RAS-GAP. We have around 30 RAS-GAPs, 30 gaps at the PSD, and this is more or less, there will be around 7 or 8 other RAS, specifically RAS-GAP at the PSD. You have different components of the gaps, family, row gaps, RAS gaps, RAP gap, that usually they have as the same as SYNGAP, both the RAS gaps and RAP gap have cross-reactivity, ARF gap, RAP gap, and the same from the GEPS domains. What their molecules are doing is they are helping the GTPase activity. So these gaps are helping to hydrolyze GTP to GTP and inactivating the RAS pathway, and the GEPS doing the opposite, just putting back the GTP and getting the activation. So mostly what we know about SYNGAP is we have some idea of how it's regulating, or at least indication that regulates ERK and some P38 pathways, but not very clear idea. When I say not clear role of upregulation, because ERK and P38 have been studied for ages at the synapse within the content of synaptic plasticity and different mechanisms of LTP. And it's not really clear what's the role of this increase in or upregulation in ERK when you have mutations in SYNGAP. You have plenty of RAS gap activity at the synapse. There are many other RAS gaps, and it to me is still not clear why, even if in the worst case scenario you lose 50% of SYNGAP activity, what is it doing with this, if you want, increase in ERK activity? You have tons of other effectors at the PSD. You have this is the GTPase families. You can see the RAS family includes the RAP family because there's a lot of crosstalk. They are included together. Many of these are present on the synapse. There are more than 150 in total in the human proteome. So what it means is that these increases in these large numbers of gaps and RAS gaps and GEPs, it may indicate you that there might be some other activities that can be regulated by changes in SYNGAP activity. We still don't know. And why we suspect also these things are because, well, for many years we were studying protein interactions at the synapse. One of these proteins that we are interested in obviously is SYNGAP, but we are doing protein interaction for many components of the PSD. Basically what we use is as immunoprecipitate SYNGAP with a specific antibody and analyze what are the interactions of SYNGAP. SYNGAP can interact with tons of different molecules. So I'm not going to describe many, but just focusing one. These large groups are GTP signaling molecules. And this other large group here are protein scaffolds. Obviously, you have a large group of kinases and glutamate receptors. So SYNGAP, it's associated, it's not just that you have many of these RAS gaps and GEPs at the PSD. SYNGAP can associate to many of them. So if this is a bit complicated graph, but it's just to have convey this idea. SYNGAP interacts with other RAS and GEPs, and other RAS gaps and RAS GEPs also interact with others. This is a common property of all these molecules. So this is just an example of 3 of them. This is AGAP2, which is an ARF gap, and this is carine, which is another abundant product, is a row gap, abundant at the PSD. You can do immunoprecipitations and protein interaction for each one of them, and we have for many other RAS gaps as well. And this is just an example of these 3, and then trying to see to describe protein interaction network for all of them. You can see in this graph with the color code that is showing you different gaps or different gaps that they are components within the same protein interaction network. This is a common property that, and even we found it in other cell types and with other molecules, that gaps and gaps, they're clustered together. And these gaps and gaps are not just RAS gaps or RAP gap, they are row gaps, row gaps. So you have a set of components all together that can be regulating the different activities within the cell. And we have many of these at the synapse, more specifically at the PSD. So obviously they are associated with the scaffold receptors, with the scaffolds and glutamate receptors, and they are probably phosphorylate. They are associated with a number of different protein kinases at the PSD, and most probably, well, they are phosphorylated, but many of them are regulating protein activity. What we know about this is that obviously is from protein kinases, we have tons of them at the PSD. We can phosphorylate any possible sequence because you have sequence specificity for any known linear motif at the PSD. You have more than 80 protein kinases at the PSD. These are a depiction of the mouse kinome. You have more than 500 kinases, and each circle is kinases that are present at the PSD. The color codes is that information that, well, it indicates what happened with that kinase after an induction of ATP, or we have data from different patterns of synaptic activity as well, if this kinase can be upregulated or downregulated. So we have a pretty good idea what is happening after synaptic activity with many of these kinases. And you can see, for example, for SYNGAP, even if we have some data of what some phosphorylation sites are doing, and mostly the KMK2 sites, it has close to, well, now it's even more, it's 100 different phosphorylation sites. Most of them have been found in brain. So there is room for regulation and for many different kinases. Not all of them are KMK2 sites or proline-directed kinases. We have a large variety of them. So this is just for to have an idea of molecular context of what will we know about regulation, how much room for improvement on finding targets are there in terms of regulation of SYNGAP activity, or what the RAS gap domain is it doing. In terms of kinases, just for this figure that we published some years ago, is just to have an idea that all of these kinases and all of these substrates, they have specificities in terms of function. So for example, you have different families of protein kinases, and they phosphorylate different families of proteins with this particular function at the PSD. Basically, if you have the CMCG group of kinases, which are mostly proline-directed kinases, they're phosphorylating mostly cytoskeleton and cell adhesion proteins, and CAM kinase families, and HAC kinase, which is protein kinase A, protein kinase B, protein kinase C, phosphorylates scaffold proteins, other kinases, and also RAS gap and gap function singles. So this is just for the end of the talk, just to have an idea what's happening here in the mature rodent synapse and what we are seeing in the human synapses. Another thing to show here is that we have a good idea as well what is happening with this component, with the core components of the protein interaction network of the PSD. They are highly phosphorylated. When you usually in all the assays that we have performing phosphoproteomics of the synapse and also other systems, usually you have, when you analyze different functions, you get like it's evenly distributed. 50% of the time you have increases in phosphorylation, and another 50% of the molecules you see decreases in phosphorylation. This is the same when you use LTP or with different patterns of synaptic activity. But the core scaffold machinery of the PSD, it has a high upregulation in phosphorylation. Usually you have increases in phosphorylation and not so much decreases. This is in general terms. And this is again to compare what we've seen in the human neurons. So in the context of SYNGAP, so we are always saying that you have a list of molecules now that with all the advances in human genetics, now these are being described even as monogenic disorders. We know that mutations in NMDA receptor, different DLGs, DLGAPs, and shanks are causing a variety of diseases with similar phenotypes, really. These are pretty, they have a lot of points in common with SYNGAPs. And so SYNGAP, we consider is a large group of synaptic proteins that are, you can take any of these, one of these, and there could be people meeting at this time together describing kids with mutations in each one of them and having this sort of phenotype and trying to address this disorder. So SYNGAP is one other of them. This is not surprising, because in our view of how we approach these signaling mechanisms, we understand that, well, it's proteins that are interacting, protein interactors, proteins that have physical interactions are more likely to be involved in the same kind of disorders, the same kind of phenotypes. So this is not unlikely. What is this when I was doing this slide is just punch me is how inefficient we had been during all these years with all the knowledge that we been accumulating, studying all of these molecules. If you ask what do we have to translate our knowledge into any therapeutics for any one of these mutations, we don't have a clue. We have been extremely inefficient. And we really, it's not just for SYNGAP, it's for any of these molecules. We can have mutations in any of these molecules. And at the moment, we are not doing anything. We cannot say, do this, and that. So in terms of that, is one of the things is that many of these things are undruggable when we come with new target, but probably is pointing out more to that we don't really understand the system after all these years. So to complicate things more, all what I show is what we know during all these years of work in mature rodent synapses. These things changes a lot during the development. We characterize these protein interactions through development, and it's very different what you get in a fetal or in E14, not fetal, but it's in the cortex of E14 mice through P7, P14, and adult. So protein interactions are very different. When I was pointing out this, probably I was going to skip this to make it in time. It's like, because we know that these protocols to induce neurons generates immature neurons. So if we have seen what the reasoning was, well, if these interactions were different in mouse cortexes through E14 to adult, what are we going to find to get really in this type of neurons? So these are just pictures how they are maturing through time, these molecules, these neurons. And I have a picture here at 3 weeks old that you can see lots of philopodia-like protrusions, and after 8 weeks old, you can start have better synapses. But everybody that works with mouse models know that these are not very few. And you can obviously you have some dendrite that you have nice many more spines, but at the end of the day, yes, you don't have as many spines as you could find in a mouse model, and these are really not much mature. So the first thing that to me is just pointing out is, okay, we are going to have these neurons. These neurons have PSDs. So we were doing looking at NCN2 neurons, derived neurons at 8 weeks old. These are some pictures that at least for us, it was nice to find PSD. You have presynaptic here with synaptic physical, postsynaptic side with the PSD there, and you can see it better here, the PSD and these neurons. We were doing mass spectrometry analysis of this, but they're showing some pictures that we have the molecules that we used to play with in the mature neurons. You can see this is soluble fraction and PSD fractions and presynaptic components, postsynaptic components. They were as they should be. You can see SYNGAP expression with different isoforms. And you have the same pattern in a fetal. This is the end of second trimester fetal brain, but you have similar patterns in a fetal brain, and I'm not showing quantitation there. But and now this is a complicated slide, but we did analysis of the different time points during development and comparing different synapses, and what clearly is showing that the protein composition of these NCN2 neurons is more fetal-like. Obviously, you have the classical pattern that you have in the fetal brain, the different number of molecules that are highly expressed, and then it being decreased through development, and the opposite pattern with adult synapses. And these changes and these differences, what mostly I'm not going to go through a lot on this slide because I want to show some findings of our own different patients. But we have seen a lot of components involving RNAs placed in these synapses in the NCN2 derived neurons.
About 5 minutes.
Oh, fine. Okay. So I try to go. So we have different. We derive different cell lines from different iPSC neurons from different patients. I wanted to show just a couple of them. This patient here with mutation, truncation mutation here, and another patient with mutation, truncation mutation there. So we have some. Well, this is what we expected or we were suspecting to have with one of these, the first model that is an haplosufficient model of SYNGAP. You have, this is the patient and this you have two corrected control. This is important for each one. You need to do the different isogenic cell line with different corrections. If you, these things are really, really, the responses are not tricky. You will need to do the different corrected cell lines. So we have increases in synaptic activity. The spike frequency, the mean spike frequency is higher. The burst per minute is also higher, but this is not what you always get. We have this with this mutation, we have the opposite result. I probably am not going to have time on what is on the specifics on this, but what I mean is that you have different mutations and you can have even opposite phenotypes and different things with the spine's morphology, the same. With the mutation in the first patient, we can see that it looks like what we define in mature spine. We can discuss later what this means in mature spines. I'm not sure that we are seeing an immature spine, but this is what we find, and it correlates what is supposed to be an immature spine. But this is a protocol that you are inducing the glutamatergic neuron. It's not just that they are just developing. So we have an increase in spine density, more the spine length and spine volume. When you look at the different mutation, it's almost not changed, and even in the spine length is the opposite. We did mass spectrometry for these were some really nice experiments. We isolated PSD for each one of the patient mutations and for each one of the corrected cell lines. So you have tons of replicates we have at least 4 or 5 replicates for each one of them and different corrected cell lines. And we obviously won't have time to describe much what are the specifics of this. Again, in protein phosphorylation, we've seen we did protein quantitation and protein phosphorylation. I'm not going to show protein quantitation, show the protein phosphorylation. It's all, again, the phenotypes show correlating with the differences of different phenotypes on different protein phosphorylation that we've seen. We have many more phosphorylation sites in increasing protein kinase activity in one of the patients, not in the other. Different types of protein domains be phosphorylated in one case and the other. We mapped to which kinases were phosphorylating those sites, and for what kinases is, to make a story short, and it's important, and just to correlate what we've seen in the, to not correlate really to what we've seen in the mature rodent synapses. We have an increased activity in kinases that phosphorylate cytoskeleton protein, which is the CMGC group of kinases, protein-directed kinases. And this is probably correlated with the increase in cytoskeletal protein activity, if you want, cytoskeleton remodeling, that are these immature synapses showing like increases, showing increasing, you have more like philopodia-like spines that are actively remodeling. We found more than 40 sites that show the same regulation in all the controls. So we have number of replicates in each corrected cell line, and we have exactly the same sign going in the same direction, which are interesting point to follow up. Some of them go in the opposite direction, and there's nice sites to look for, sites that we study in the context of mature synapses as well, AMPA receptor auxiliary units, calcium potassium channels, solar gaps, protein kinases. So these are interesting things to show. So probably I won't have time to show all the data, but we mapped with the protein interaction data in these models as well, which sites were upregulated, downregulated with each mutation within the SYNGAP protein interactors. And I'm not going to show anything related to brain organoids, but we got SYNGAP expression in brain organoids, also the interactors there, and also we derive inhibitory neurons from these patients as well. This is SYNGAP expression, immunoprecipitation from, these are specifically somatostatin neurons. So what we are doing is combination of inhibitory and excitatory neuron. The dish is showing one mutant, one wild type, both mutant with one wild type, and also we have other cells there. So probably it's not much time, but what we really need to is just to convey the idea. I want you that the mutations, even if some of them, and I agree with what has been said before, we have this disruption of SYNGAP activity. What do we know is to what extent? I'm not sure that all the hapless deficient models is like 50% decrease on SYNGAP levels. We are going to have many more phenotypes and also patients' phenotypes to what the mutations are really doing. So if we are considering that most of them are just some function and haplosufficient model, we are going to find lots of different phenotypes, at least in our models, that are not so well correlated. So I feel that we need to have a better way to characterize really properly what each mutation is doing to the SYNGAP protein. And we are working on that. And then most of this catalog of things that we were seeing is just to be clear, what do we need to look for? One of the, obviously, when we are trying to improve the levels of SYNGAP, is just to look for SYNGAP. So in that regard, we need the model to determine properly how much SYNGAP do we have. And also, if we are looking for phenotype, which ones are we looking on in which model? And that's more related also to if there's room to find other targets as well. So those are collaborators fundings, and mostly I would like to have here the SYNGAP Research Fund and the SYNGAP family, because the work that they're doing, I always say it's great. And in my case, I was working with other molecules at the PSD all the time, and it's just helped me to reconnect with SYNGAP from my early years of postdoc, really. Thank you.
Great. Thank you so much, Marcelo. That was fantastic and a whirlwind tour. There's some discussion in the chat, and I would encourage you to look at some of that and continue some of that discussion in the chat box, so that we can move on to our next speakers. I think we're going to have a